Okay. Okay. Can someone please confirm whether you can see the slides or not? Then I will begin it. Please write it in the chat box whether the slide is visible to all of you or not. Yes, okay, fine. So we can start today's session. So for week one, the very first thing that you all of you have to do that is remember the structures of the amino acid. For this course or this biochemistry course, you have to do this thing. Okay, so there is no other way, no way you can I mean, if you do not remember the structures, then you won't be able to solve the questions of any week. I mean, these are the primary or the basics thing that you will be needing. So I have put this slide for all of you in case in some question we do need the help of the structures so that I can show you. So the first thing is you need to remember their structures, their name, their 3-letter code and 1-letter code. This is the very basic thing that we will be needing to remember throughout the course. Okay. So I am just showing this. This is the basic thing that will be required. So we will begin with our first question. So it states, which of the following single letter codes represents the structure below? So what we can see, it is a peptide, right? So various different number of amino acids, I mean 1, 2, 3, 4, 5, 6, yeah. 6 amino acids are attached to each other via peptide bonds. So the structure is shown here and you have to identify the single letter code for this particular peptide. Okay. So I will give you like 1 or 2 minutes to solve it, and then I will check the chat box for your answers, and then we will discuss it. Okay. First try to solve it by yourself, and obviously as I said you will be required to remember the structure of amino acid to solve this. And make sure that you do clear every doubt you have in these preliminary sessions and weeks, because these basic things will be very much required when we will be moving to the uh, next I mean the latter weeks assignments and everything, because the more the time progresses the course will be a bit difficult than this. Okay, so the make sure you clear all of your doubts in these basic sessions. If anybody has solved it, they can write the correct option in the chat box. Okay, I have one answer. I would like everyone to try it, because here we are trying to collectively solve the questions. Even if you answer it wrong, it doesn't matter, because we are here to correct whatever doubts we have. We are here to clear that. So I will request all of you to try at least. It doesn't matter whether it is correct or not correct, but at least try to answer the questions. Okay. So far I have only one answer. At least I will wait for another person to answer this question and then I will show you the answer and discuss it. At least another one. I can see there are almost 32 people, 31 to 32 people who have joined this. So at least 2 to 3 people I can expect that they will be able to answer this particular question. Okay. Uh, who is raising hand, you can write your query in the chat box. Okay, I am watching it constantly, so you can just write it down. Okay, so I have two, I can see two things, two option. One is option A and another is option D. So okay, so let us look at the correct answer. So the correct answer was option A. So how we are doing it? So basically whenever you see a question like this, so as I have already said, okay, so first of all I would request all of you to mute yourself, otherwise it becomes a bit difficult for me. Okay, so please I will request any question you can write it in the chat box. Okay, so back to the discussion. So to solve this kind of question, we need to find, so basically this is a peptide structure. Peptide means the proteins or the basic amino acids, they are actually joined to each other via peptide bond, that is nothing but this amide bonds that are there. Okay, so this is the basic thing of a peptide. So via amide bonds they are linked to each other, this is the basic thing. So now first we have to identify the amide bonds and then we have to break the structure along the amide bonds to get to see that what amino acids are present there. Okay, so the first amino acid that amide bond that was there was this particular one second. So this is our first amide bond that is present. So now we have to break the structure along this amide bond. If we break it, what we can get? On the left hand side it will be COOH and on the right hand side it will be NH2 or NH3 plus. We can write it like that. So if now say one second, let me turn it to pen. So now we have our structure limited to this. So this is our structure NH3 plus. After breaking this will be COO minus or COOH, anything you can write, and this is basically your first structure. So what amino acid is this? NH2, NH2C COOH, and there is one CH3. So basically it is alanine. So what is the single letter code for alanine? That is A. So the first one will be A. Similarly, we have to move to the second amide bond and break it there. Again, if we break it here, so here we are getting, suppose NH3 plus. So now if we break it here, we will generate a COO minus here and a NH3 plus, say here. So now our second structure becomes something like this. So what is this structure? NH2, CH2, COOH, and in the R group it has CH2OH. So this particular structure is nothing but serine. So the next single letter code for serine is S. So obviously the second one becomes serine. Okay. So up to that both option A and D are same. I have seen there are two options that you have selected, A and D. Next bond, amide bond, is here. Again similarly we have to break this here. After breaking, the structure that we will be getting is NH2 CH2 COOH. No substitution here. Only one amino acid have this, that is glycine. So up to this both option A and D are same. After that the variation starts. Now let us move to the next amide bond. Here after breaking, what we can see? It is NH2CH2 COOH and the side chain is pH sorry, CH2PHOH. That is the structure for tyrosine. Now for tyrosine, the single letter code is Y. So the wrong that whoever got it wrong, that is tyrosine and phenylalanine. Phenylalanine was basically like CH2PH, but it is tyrosine because it has CH2PHOH. Okay, so that is where you got it wrong. Next amide bond is this one. So we will get an amino acid of this particular one. So again the side chain is this. Obviously this particular side chain exists in only one amino acid, that is valine, and the single letter code for valine is V. So the next one will be V, and the last amino acid, that is here, and here we can see the side chain is CH2 COO minus. And for CH2 COO minus, one second, aspartic acid, CH2 COO minus, uh, one second, one second, one second, my pen is not working. So the last one is for aspartic acid, right? So this is the structure for aspartic acid, that is CH2COOH, and for our case also the R group was CH2COOH. So for the single letter code for aspartic acid will be D. So our correct answer was ASGYVD. So all that you need to do is to break the amide bonds and then identify the R groups and their corresponding amino acids, and you also have to remember their one letter code. Okay, so I hope there is no doubt in this question. Moving to the next one. So this is your second question. Try to answer this as well. It states the process by which information encoded in a gene results in the formation of a functional product in the form of a protein. So basically from a gene to protein. This is called gene expression. How it is formed from gene to the protein that is being formed. The gene expression pathway can be represented by one of these four options given below. You have to select the correct option. Okay, I have already three answers. Thank you very much for that. Okay, I think everybody is very clear for that. Fine. So the correct answer was option B. I would not waste more time in this question because everyone got it correct. For even if somebody has any doubt, so the basic process is, the first process is from gene to protein. First what you need to do, one second. First, what you need to do, you have your DNA. So, first of all, if you want to know what information is there, first you need to break the double strand basically. So, that process from like DNA to RNA or mRNA transformation, that particular process is known as transcription. And once you have your mRNA, all the information about the amino acids and proteins that are there, all the information that was there have to be transformed via this mRNA, which is called translation. And according to that, whenever it will reach to the ribosome or something, by gaining those information, it will start forming the proteins and uh, forming the sequence of proteins accordingly. So this is the basic process that where gene expression occurs. First transcription, that is basically from double strand to single strand. Then this mRNA actually takes the information, that is called takes this information to the ribosome, that is basically called translation, and via which then the protein sequences will start forming. Okay. This is your third question. Okay, so I think all of you also know this. So it states amino acids can be classified according to polarity and polarity of side chains. From the options below, choose the one which contains amino acids with only aromatic side chains. Okay, so we can actually divide amino acids in different types like acidic amino acids, basic amino acids, polar and non-polar amino acids, also aromatic amino acids. Everything of this depends on the structure of the side chain basically, because the main structure, that is NH2CH2COOH, it is same for everyone. So only depending on the structure of the side chain, that is R, you can classify them according to their polarity or aromaticity, etc. Okay, so in which of this option all of the amino acid can be classified as aromatic side chains? Okay, I have many answers. Thank you very much for answering. Again, this is a very important question because this is very common, because everywhere it is asked which are the aromatic side chain containing amino acid. There are only three in the 20 essential amino acids. There are only three, and the correct option was D. They are phenylalanine, tryptophan, and tyrosine. These three are the aromatic side chain containing amino acids. And why they are very important is that I think ma'am, Professor Dasgupta has already discussed in her lecture. That is, these aromatic amino acids are actually fluorescent. Okay, so that is the huge importance of them. That's why we classify them differently, because we can identify them from fluorescence. So whenever any protein is showing some kind of protein or peptide is showing some kind of fluorescence, we can assume that they have this aromatic side chain containing amino acid. So there are only three. This helps us in narrowing down the possibility of amino acids whenever we see the fluorescence. Okay. Sorry. Let us move to the next one. This is very easy. I will not even ask all of you to answer it. I'll just directly show you the answer of this. So the isoelectric point of an amino acid is basically defined as the pH where the molecule carries no net electric charge. So basically it does not means that the entity itself is uncharged. So this is, sometimes I have seen students make this mistake. They assume as the in the isoelectric point, as the net charge we say it is 0, they assume then the entity itself uncharged. No, that is not. Say suppose for glycine, in its at its isoelectric point it is remaining as NH3 plus CH2 COO minus. That does not mean the entity is uncharged. It has charged species like NH3 plus and COO minus, but the overall charge, the net electric charge, is 0. So be clear with that. Okay, do not mix it with like the entity is itself uncharged species. Okay. Moving to the next one, I would like all of you to solve this. That is, a buffer is prepared with 1 molar HA and 1 molar NaA. HA is the acid and sodium A, that is NaA is written, is the salt. So pKa of HA is 4.87. It is already mentioned. What will be the pH of the solution? Okay, I have one answer. Okay, I have two answers. For others, I can give you one hint. You have to use the Henderson-Hasselbalch equation. Okay, the basic equation for Henderson equation that we use for calculating pH of a solution containing salt and acid. Anybody else has got the answer? I can see two answers. Others? Okay. Another one is saying answer B. Okay. So I can see three answers. Let us see the correct one. So the correct one should be the option A. How? So this is the Henderson-Hasselbalch equation that I was talking about. So basically this is your equation. pH equals to pKa plus log of, this is your salt concentration or the base concentration you can say, and this is your acid concentration. So this is the basic structure, sorry, basic equation. So all you need to do is to put the concentration values for NaA and HM. Both are 1 molar. So obviously this will cancel out. So pH will be equal to pK. As the concentration of Na or A minus and Ha are same in this particular case, so pH will be equals to pK. So our correct option would be 4.87. Okay, moving to the next one. Fine. This states gamma carboxyglutamate O, O-phosphoserine and O-phosphotyrosine are a few enzymatic modifications of amino acids that form after protein biosynthesis. So the important point here is after protein biosynthesis, the enzyme modification is happening. There are very important components of cell signaling pathways. This kind of modifications are known as what kind of modification? I mean, what is their name basically? So the main point here is the modifications are occurring after the biosynthesis of protein has been done. So what do you call this kind of modification? Okay, I have options like this. So obviously, uh, ma'am has already discussed this in your class. So the correct option for this, yes, everyone is correct. Thank you for answering. So the correct answer is post-translational modification. So obviously, if the protein is being biosynthesized, then you have to reach to the last step. Okay, so you have to skip the transcription because transcription is the middle step, like from DNA to RNA, and then during translation the mRNA will give the all the signals or all the ideas that what amino acids you need to synthesize for this protein formation will be done in the translation process. So if protein has been synthesized, that means translation has already happened. So you do have the knowledge of the amino acids that you require for the protein synthesis. So if translation has already been happened, so obviously the method will be called post-translational modification, because after protein formation you are doing that. Okay, very good. So moving to the next one. Okay, the greatest buffering capacity at physiological pH would be provided by a protein rich in which of the following amino acid? So you are trying to use it as a buffer which have buffering capacity, a protein. So which amino acid should it contain mostly to have this buffering capacity at physiological pH? So the first thing that is important here is it is state. Mm, I would request again to all of you, please mute yourself, otherwise it becomes, I mean, too much sound comes, so it becomes a bit confusing for me. So it's a request to all of you. Okay. Moving back to the discussion. So it was our question. So which of these following amino acids would be the correct option to act as a buffer in case of physiological pH? I have one answer, two, three, four. Okay, very good. So basically, they are you all of you are right. The correct answer will be histidine because the physiological pH is 7.4. And how do you understand which one will be the I mean the ideal one? Okay. So to understand that, what you need to do is to look at their PI values. That at what PI values they will be remaining neutral. Buffer capacity means something that will not try to change the pH of your solution or the stop the change of the solution's pH. So basically if you look at their basically if you look at their PI values, serine, cysteine, alanine all have PI values out of the physiological range like 5.70, 5.15 and 6.11. Only histidine is close enough to physiological pH. I think my mouse is malfunctioning. Very sorry for that. Okay. So only histidine has a PI value that is close enough to the physiological pH. Okay. It is malfunctioning too much. Looks but let me try it once again. Okay, so the correct physiological pH is 7.4. So we can see only histidine uh, uh, passes through this criteria. So histidine will be the correct option for choosing a protein that will act as a buffer in the physiological pH. Okay, fine. Okay, this is a very interesting question again. So it states some proteins contain additional amino acids that arise by modification of amino acid already present in the peptide, like we already know about post translational modification. So sometime amino acids will get modified after the proteins are formed. So basically they are asking which of the following are examples of modified amino acid residues. You already know about post translational modification, and after that, out of these three, oh, this is very much problematic. Sorry for this again. Okay, so out of these three, which are modified amino acid residues? This is the question. Select the correct option and state in the chat box. Okay, fine. Second and third one. So you are basically choosing the option D. Okay. Others? What about others? What do others think out of these three? Which are the modified amino acids? I got another one. This says option A. Any third opinion? Because now the options that I have got is option A and option D. So any third opinion who agrees with either option A or D, or they can have their own option as well. They can either select B or C as well. Option D. So mostly they are talking about option D. Okay, fine. So the correct answer for this should be option C. Okay. So if I have to answer this particular question, you actually do not need to know the structures of the first two, because you already know that the essential, in case of essential amino acid, there are two amino acids, that is proline and lysine. So basically these two are modification of proline and lysine, 4 hydroxy proline and 5 hydroxy lysine. So basically they are the modifications of a important basic amino acids. Okay, so these two options are obviously correct. They are modified amino acid residues. They already have the names of the amino acids in them. I thought what you all will be having the problem with is the third option, that is GABA or gamma aminobutyric acid. It is a very important compound in our body. So basically GABA does not matches with any of the amino acids, so 20 essential amino acids we know. The name at least does not match with the essential amino acids that we already know. So basically if we look at the structure of GABA, it will be 1, 2, 3. If this is NH2, my handwriting is very bad. I am not much used to write with mouse and I don't have a stylo pen, so sorry for that. So basically this is the structure: 1, 2, 3, 3 CH2 groups, and at one side there will be NH2 and there will be COOH. So basically alpha, beta, gamma, gamma aminobutyric acid. So this is your alpha, beta and gamma carbon, and so the name is gamma aminobutyric acid. 4 carbons, so it is butyric acid obviously. So where does it comes from? So does this resembles to any of the essential amino acid structure? If you see it, if I write it down again, suppose it is NH2, NH2, it is COOH. Again, sorry for this horrible handwriting. Okay, now if I just add another COOH group here, does this resemble to any of the essential amino acid that you already know, right? So what is the structure? NH2CH2COOH, and this is your R group. This is CH2CH2COOH. It is nothing but your glutamic acid. You have already read this. So basically the GABA or gamma aminobutyric acid forms, actually is the modification of glutamic acid. There is some enzymes called like glutamate decarboxylase. So from the name you can already guess that basically decarboxylates means that you are eliminating the COOH group. So this glutamate decarboxylase, what it does is simply eliminates this particular carboxyl groups and gives rise to GABA. Okay. So I thought this will be the option that you might find a bit difficult to choose from. So this is the basic thing. So for the correct answer, all of these three are the modifications of amino acids. Okay, so I hope this particular question is clear. With this, let's move to the next one. Okay, it states which of the amino acids below is the uncharged derivative of an acidic amino acid. So the points that are important, that is uncharged derivative of an acidic amino acid. I think all of you will be able to answer this. Quickly write it down in the chat box. Okay, I have already got an answer, C. Okay, so thank you for answering. Okay, so the correct answer is C. It is very obvious because if you look, cysteine, tyrosine, serine, none of them are like acidic amino acid. Acidic amino acids means where you have an additional COOH groups basically. So there are glutamic acids and aspartic acids. Those are the acidic amino acid. So once in case of glutamic acid, the R group is CH2CH2COOH, and if you remove the COOH group and replace with it with a CONH2, then it simply becomes glutamine. And as you now the structure, if I see, so you have your NH2 COOH, NH2 COOH, NH2 COOH, and this is your CH2CH2 COOH. This is basically glutamic acid, that is your acidic amino acid. And under acidic condition, this hydrogen will be removed, this will be COO minus, this will be COO minus, and this will be NH3 plus. So 1 plus and minus charge will be cancelling out, but one negative charge will still remain. But if you simply replace with CH2 CH2 CONH2, so now no longer this deprotonation needs to be occurred. So basically the only thing deprotonation and protonation will occur in this NH2 and COOH. One will be very sorry, one will be NH3 plus and another will be COO minus. So overall it will be an uncharged species. So the correct option for this one was glutamine. I hope this particular question is clear to you. Sorry, let us move to the next one. Okay, it states the hydrophobicity scales are values that define the relative hydrophobicity or hydrophilicity of amino acid residues. It is used to characterize or identify the possible structure or domains of a proteins. You already know there is a hydrophobicity scale by which we can identify whether the side chains are basically, because the rest of it is same for all the amino acids, only the side chains that varies. So in case of side chains, whether according to these values, whether they will be hydrophobic or hydrophilic. Okay, so these Kite-Dulite plots are given below. The plot has amino acid residues on its x-axis and degree of hydrophobicity or hydrophilicity on its y-axis. It is already written. One second. Okay. So now they are asking pointer. So this is the sequence ARNDCQ. This is your full sequence ARNDCQ. So their corresponding value are also given and the Kite-Dulite plots are given. You have to identify which among them is the correct Kite-Dulite plot for this particular sequence. I have one answer, that is A. Okay. Okay. two answers I have got. Both are option A.
Admission code is 100% going inside the hall, C block, canteen. Admission code is 100% going inside the hall, C block, ground floor, canteen.
Okay. Okay, thank you very much for answering the questions. So most of you have answered selected the correct option, that is A. So let us look at, oh yes, the correct option for this question was A. So basically this question is very easy. All the values are given. What values you have to put in y-axis and x-axis, all are given. Only the trick is that whether you remember the one letter code of the amino acids or not. Just you need to understand that alanines means A, arginines means R, N is for asparagine, aspartic acid for D, C is for cysteine and Q is for glutamine. Thus, just this thing you need to understand, nothing else. So let us see the graphs. Let us start with this option A. So alanine is 1.8, obviously it is positive. So it is correct. Arginine is minus 4.5, again in negative, and it is correct. Asparagine and aspartic acid both are minus 3.5. Okay, so N and D both are minus 3.5. So let us see. This has increased from minus 4.5 to minus 3.5. So it is again correct and both are same. So these both are correct. Okay. Next for cysteine it is 2.5. So I have to move to the positive one. So 2.5 it is correct and it is higher than 1.8. So again it is correct. And the last one is Q, that is minus 3.5 again for glutamine. So again we have to move back to minus 3.5. So just you have to check whether this matches with the graphs or not. One thing that can happen in case of your assignment or exam, in this case you can see ARNDCQ. This is in this form the peptide is sequence is written, and also the listing is given is, like that as well in the same sequence like ARNDCQ. But in exam this might not be like that, to test whether you actually remember the one letter codes or not. This might be shuffled there, okay, or in the assignments as well. Very good. So let us move to the next question. Okay, this is a very easy one. I won't be showing you the options in this case. I want to all of you to write down the code directly. So basically they are asking for the correct one letter code of this tripeptide. That is three letter code is given there. I'm not going to tell their names even. That is ASN, TYR and LYS. What will be the one letter code for this tripeptide? Okay, I can see one answer. Okay, she has corrected it to NYK. Another answer is NYL. Okay. Another answer NYK. Okay. So, what is ASN? It is asparagine. TYR is nothing but tyrosine and LYS is nothing but lysine. Asparagine, everyone chose the correct option, that is N. For tyrosine, it is Y, that is again correct. I think the only mistake that you could have made, that is about lysine. Lysine, sometimes when you chose L, that is not the code for lysine. L is the code for leucine, not lysine. For lysine, the one letter code is K. So the correct answer should be NYK. Okay. Thank you very much for answering. Many of you, most of you has answered it correctly. Okay. This is again a very basic question. It will not even require much time for all of you to answer. So a dipeptide basically has how many amino acids and how many peptide bonds? This is generally it is asking. Okay. We all know that dipeptide means there are two amino acids and one peptide bond. Do not get confused with the name. that dipeptide means there will be two peptide bonds. No, di means diamino acids are joined with each other via peptide bonds. So dipeptide bond means there will be two amino acids and they will be joined by obviously one peptide bond. Okay. Moving to the next one. Okay, this is a very interesting question. I would like all of you to read it um correctly. So, this is the representation of this Nobel winning experiment. You all know that. So, this is your RNase A, ribonuclease A. It has 8 cysteine residues and thus has this 4 SS bonds. 2 cysteine residues will form one SS bond. So basically 8 cysteine residues. So there will be this 4 cysteine bonds. So this was the experiment. First, it is the native structure of the protein and we all know only at its native state it will be catalytically active. Okay, so after the addition of urea and beta mercaptoethanol, we get this kind of structure. Now you have to identify in case of A whether it is native or not, and in case of B whether this structure will be catalytically active or not. Let's look at the next step. In the next step, they removed urea and beta mercaptoethanol. Both of them are removed. Now in case of X and Y, again you have to choose that whether it will be, in case of X, whether it will be native or not, and in case of Y, whether it will be catalytically active or not. This is the option you need to choose. So this is your representation and these are your options. Read the options properly and then choose the correct option. Okay, take your time to read the options properly and then answer in the chat box. And could you show the questions? Okay, so let me repeat tell you the question. What is the question? So you can see there are four things written here: A, B, X, Y. Look at their colors. In the first case, it is written in black color that it is native, and in green color it is written as catalytically active. So in the second structure where there is written A, you have to identify whether it is again native or not. For option A, you have to identify whether it is a native structure, this particular structure is native or denatured. And in case of B, you have to identify whether it is catalytically active or non-active basically. And then there is other catch lines there that I will discuss later. Similarly for the third structure as well, in case of X, you have to identify whether this structure is native or denatured, and in case of Y, whether it is catalytically active or inactive. Once you find these four things, then there will be a catch about these disulfide bonds and everything that is written there. First, just try to identify whether A will be native or denatured and B will be catalytically active or not. Similarly, X and Y. First identify those and then narrow down your options and then look at the options and picture and select one of them. Just by identifying their native or denatured state, that is A and X, and their catalytic activity, you can already narrow it down to two options, I can say it. And after that you have to look at the picture very carefully to understand what is the state of the proteins. Okay, so some say option D, some are saying option B. Okay, thank you for answering the question. So let us look at the solution. So first we need to identify whether the second structure for this, it is native or denatured. So obviously we can say that it is a randomly coiled structure. That is not a native structure. Okay, so it is basically a denatured structure. And for B, if the protein is denatured, so obviously it will not be able to act as a catalytically active species. So the correct option for A and B is it is denatured and catalytically inactive, because it is not its native state in its native state, so it will be catalytically inactive. So let us first find these things. In this case, it states A is natured. So this option is wrong. Okay. A is not native. For this, it is written as A. A is not native. So this option will not be correct. Next, say A is denatured and B is catalytically inactive. So this option for now justifies the things that we are saying. That is still matches with the options that we are saying. So we will still consider B. Next, look at this. Again it says that it is a native structure. We know it is not a native structure. So again we can rule out this particular option. Look at the fourth option. It states that this species is denatured and catalytically inactive. Very good. So again we will stick to this option as well. Now let us move to the X and Y. What does it say? You can already see that the first structure and the third structure are same. Why am I saying they are same? Because not only they are coiling, this is just a pictorial representation. Obviously in reality it is not that simple, but from the pictorial representation you can already see that the coiling that is there looks exactly same, and not only that, the position of the disulfide bonds are exactly same as well. So we can say that the third structure sorry, is exactly similar to the first one. So our X is again native. It has become native, and it is obvious because you have removed urea and beta mercaptoethanol. So it has again become a native structure. And if it has become native again, so again it will be catalytically active. So let us see. It states, sorry, this option is not we are considering. So it is stating X is native and Y is catalytically active. Very good. Here also it is stating X is native and catalytically active. So by only seeing A, B and XY, we still cannot choose which of these two options will be the ultimate answer. So let us take the hints from the other words. The first thing says the disulfide, in case of this second structure, the disulfide cross links are reduced to cysteine residues. Okay. In the D option, it states for these structures, disulfide cross links oxidize to yield cysteine residues. Okay. So see, first of all, the problem is, so this is your disulfide bond. If you have to break a disulfide bond, you have to reduce it, because disulfide bonds are formed by oxidation of SH bonds, right? They are getting oxidized and forming SH bonds, sorry, SS bond. So basically, if you have to break a disulfide bond, you have to reduce it. So the term that is written here, that the disulfide crosslinks are reduced to yield cysteine residues, that is SH residues basically, because cysteine has this CH2SH as the R group. So this particular thing is correct. But here it is says the disulfide crosslinks are oxidized. So this term is not correct obviously, because you cannot oxidize a disulfide bond to a SH1. That is a reduction process. So already we know that option D will be not correct. Yet let us look at the last portion of the this sentence as well, because this will further help you in solving another question in this session as well. So the last portion says, in the fourth structure, that is X and Y, disulfide crosslinks are reformed correctly. Okay, so in option B it has saying, when the disulfide bonds are forming, they are formed correctly. In this case it is saying disulfide crosslink reformed randomly. But you can already see from the pictorial representation, then it is exactly matching the native structure. So you cannot say that the disulfide bonds has reformed randomly, right? Because if it was random, then the positions were not same to the initial one. But we can already say they are exactly same. So obviously the disulfide crosslinks are reformed correctly. So this is the correct statement from every way. So our correct option should be B. Okay. Why I am discussing all aspect of this all these options, because in exam or in your assignment, it might be that only one thing might be changed in the option. Like here we can already say there are two things that are different in options B and D. But suppose in your assignment or exam there is only one small difference is there. Like only say three things are same, only they are saying the disulfide crosslinks are deformed correctly, and here they are saying randomly. Only by seeing these things you have to identify. In case of that, you have to understand what is happening. That's why I explained it elaborately. Okay. Okay. So I hope this thing is clear to you. Moving to the next one. Okay. Which of the following representations of cysteine is not in its naturally occurring L form? So these are the four form. All these structures are in flying wedge representation, and you have to identify which of them are not in L form. L form is the naturally occurring one always. So which of them is not L? So obviously it is not L, then it will be D. I can hope that you people already, all of you already have a knowledge of stereochemistry, because otherwise you won't be able to solve this particular question, because you must know how to convert it from flying wedge to Fisher projection. Only then you will be able to identify it. If you do not have the idea of the stereochemistry, then you can write in the chat box, because in that case I will skip this particular question. If you know, you can solve, and if you do not know, you can write it down in the chat box, because then in that case I will skip it, because that is a stereochemistry is a totally different basis or totally different subject. So basically you need to transform from flying wedge to Fisher projection and then see what is the correct option. So while determining D or L, one thing you must remember, see if this is your Fisher projection, that you place your C double O minus in the upward, your R group in the down. So the whole carbon chain is in the vertical position and C double O minus is up. Once you get to that, in your Fisher projection, if the NH3 plus is on the left side, then we call it L form, and if the NH3 plus is on the right side, then we call it the D form. This is the basic thing. So while converting, you might have to make further changes to present the structure like this, because only then you can identify whether it is L and D. It is very crucial that you place C double O minus at the top and R at the bottom, so the carbon chain stays at the vertical chain, and then you place your NH3 plus and the hydrogen group. Okay, I can see one answer that is stating B. Anyone else who has solved it? If you do not know, then I will insist all of you to learn it from any YouTube videos or anywhere, or any book where stereochemistry is mentioned, because you will need this idea. The basic conversion from flying wedge to Fisher, you will need this. Okay, so I can see two options, but they are different. One is B and another is D. So let me wait for another answer at least. Then we can see who is the correct one. Okay. So most of you are saying B, but I have to say the correct answer is D. Make sure you have placed the Fisher projection like this, that is C double O minus is upward and R is downward, and then accordingly after placing them, whether the NH3 plus is on the left or on the right. So the correct option should be D. Those who have answered B or selected option B, you might have done something while exchanging the groups. So if you still have the doubt and cannot find the correct answer, you can send an email uh, to myself directly or in the NPTEL team stating my name. I can share sorry, I can share the solution with you. Okay. If you still cannot figure out why it is D but not B, you can send a mail stating my name. I will share the solution because I cannot particularly start a stereochemistry session here. It will not be possible, but I can share the solution via mail. Okay. The correct option should be B. Sorry, D. No, the B will not be that. B is option B is not a, B is not correct. Okay. Let us move to the next one. Those who all have doubt in the stereochemistry part, please send an email. Then I will share this particular solution with you via email. Okay. Those who have selected the correct option D, they are fine. But those who are finding it problematic, I will share the solution with them. Okay. So fine. This is your next question. After this, we will take a short break of like 5 to 6 minutes and then I'll be back. Okay. So this question states an mRNA molecule synthesized from a DNA template strand have the following sequence. So the sequence is 5 prime and this, then this, and then end with 3 prime. What will be the its complementary sequence? So what will be the complementary? So this is one strand that is given here. So what will be the sequence of its complementary strand? That is basically they are asking. But make sure it is the strand is given from mRNA molecule. So make sure to consider that as well. Okay, so we have the answers. Options A. Thank you for answering. Again, so first of all, from a DNA, we are getting an mRNA. So basically, from DNA to RNA means you are splitting it from the middle. So whenever you are splitting it from the middle, you know that in the opposite sides, you always have the complementary bases. That for A, the complementary base for DNA will be T, and for G, it will be C. So if you just think about that, so what should be the complementary bases? So that should be T, that should be A. This particular G complementary should be C, this one should be G. For C again will be G, for A this should be T, for G it will be C, for A it will be T, and for T it will be A. Okay. And one strand goes from, if it goes from 5 prime to 3 prime, then another one will go from like this, that is 3 prime to 5 prime. That is the basic structures for DNA. So if it is 5 prime and 3 prime, then the complementary strand will be 3 prime to 5 prime. So this is the structure of the complementary strand. But it case it is saying an RNA is being synthesized. So whenever an RNA is being synthesized, we all know in case of RNA the T's are replaced with uracils or U. So wherever you see T, that will be get replaced with U. So your correct answer should be option A, that is 3 prime U. This thymine will be replaced by uracil. Then A, C, G, G. This thymine will also be replaced with uracil. Then C, again this will be replaced with uracil. A, 5 prime. Okay. Okay, so let us take a 5-minute break. Then we will get back to the question. I will be back at around 8:25 or 7:25 or 7:26. Okay. Okay, so let us restart the session. I hope the presentation is visible to everyone. It should be, I can see it. Okay, so let's start with the session again. And let's move to the our next question. Start presentation. So this is our next question. It states many antibiotics interfere with the transfer of genetic information from RNA to protein, preventing bacteria from growing. Which of the following process is thus affected by these antibiotics? So this is just states a particular one of the mechanisms of antibiotics to work is they interfere with the transfer of the genetic information from RNA to protein, like from mRNA to protein basically. So which of the following processes will be affected by the antibiotics? It is basically written in the questions as well. You just need to understand. Okay. I have one answer. Okay. Two I can see. Okay. Okay. So yes, the correct option for this will be option B. Obviously, because for like bacteria to grow, you have to transfer the information, because whenever you are trying to replicate, so bacteria, what they will try to do basically, they will try to replicate themselves. So whenever they are doing that, to replicate something, you will always need to know the informations that are in the gene, and only if you pass it and only if you get the information, basic information, like what are, say suppose in the bacterial DNA or something, whatever the amino acids that are present, only if it gets that information, it can then start synthesizing the peptides, the required peptides, and the following proteins. Okay, so if it does not get to do that, then their functionality or the replication will be hampered basically. So it is basically stating it is hampering the information transfer step from RNA to protein. So which RNA basically does that? That is basically your mRNA, that is transform the information to protein. So that particular step is all obviously known as translation. So this particular mechanistic pathway of antibiotics affects the translation process. That is what was asked in this question. Moving to the next one. Okay. Three buffers are made by combining a 1 molar solution of acetic acid with 1 molar solution of sodium acetate. So basically it is your HA and Na in three ratios. Okay, so there are three buffers, buffer 1, 2, 3. One second. Okay, so buffer 1 have 10 ml of 1 molar acetic acid, 90 ml 1 molar sodium acetate. Buffer 2 has 50 ml 1 molar acetic acid and 1 molar 50 ml 1 molar sodium acetate. Buffer 3 has 90 ml acetic acid 1 molar and 10 ml sodium acetate 1 molar. Okay. So which of these statements is true for the resulting buffers? So this is the buffers pH sequence, or this or this or this. These are the four options. Choose from this correct option from them. Okay, I got one answer. Okay, another answer. So most of you are saying option A, and one of you have said option A or D. So one thing I can confirm you, do not require actually to know the pKa value, because in all of these cases the pKa value will be same, right? Because they are all the mixtures of acetic acid and sodium acetate. So the pKa value for acetic acid will be used in this case. So pKa value is just same. So just by comparing the other part of the Henderson equation, like pKa of log of NaA by HA, we just by comparing that part we can answer this question. So you can uh, rule out the fourth option. So you do not require actually to know the pK, because you just need to identify the pH values, not the exact value. So the correct answer for this is option C actually. I think one of the thing that all of you understood, that obviously the pH of the buffer would be different in all cases. But I think just in case of whether the pH will be higher or lower, that you all got confused and most of you answered A. Even actually you do not require to do any mathematics or understand any mathematics for to solve this particular question, because in all these cases you can see 10 ml and 90 ml, 50 ml, 50 ml and 90 10. So in all cases the overall volume, I mean the final volume of the buffer is fixed. Whatever you are doing is just varying the salt concentration and acid concentration. So salt is nothing but basically your base concentration, and this is your acid concentration. Whenever you increase and the acid salt, that's we get in case of that, this is your acid salt or the acid in this case. Whenever you increase the acid part in a buffer with a fixed volume, an acid part means it will give more acidic conditions to the buffer. That means the pH of that particular thing will start decreasing. Like if we do take example of the like normal phosphate buffer, something like that, like a potassium dihydrogen phosphate and dipotassium hydrogen phosphate. In both the cases, if you start increasing the acidic salt or the acid part more, then the pH start decreasing, because generally you are increasing the acid part, nothing but that. So obviously in the third case, in case of buffer 3, you are using 90 ml of acetic acid, that is out of that, I mean 90 into its molarity is 1 divided by 100. This is the concentration of your acid part, and upwards it is 10 into 1 divided by 100. So obviously in the third case it has the most acidic part involved in the buffer. Even if you do the calculation, you will get to the same conclusion. So if it has the maximum amount of acid part, so the pH should be less for that. So because we know for acids the pH is less, for basic the pH is higher. So the third option is the correct. I think you all understood it correctly. Just got it wrong in the last part, because it has more acid. More acid means the pH should decrease. So the correct option will be option C, where the pH of the buffer 3 will be minimum, because it has the maximum acid part present in your system. Okay, I hope it is clear. Let us move to the next one. Okay, so it states of the 20 standard amino acids or the essential amino acids, basically only dash, there is a dash, is not optically active. The reason is that its side chain is what? It is a very easy question. Which of them is optically inactive and why? I already have two answers. Yes, I think all of you already know the correct option should be glycine. That is very easy for any molecule to be optically active. So basically it has to bend the light to some degree. So for that the molecule has to be like chiral. But in this case of glycine, we can see this is NH2, this is COOH. I'm just writing simply. There is one hydrogen and this is your R group. Generally, for all other amino acids, this R group is different than hydrogen or this NH2 and COOH. So in all the cases, the four substituences are different, that is all of them are chiral. But in case of glycine, this R is hydrogen. That means two of the substituents are similar. So whenever you are going to pass light through this molecule, one part of the molecule will rotate the light to whatever extent, the other part will cancel it out basically. Sorry, I have write it down oppositely. Suppose this is your structure. You have hydrogen, hydrogen, yes, NH2, COOH. So there is the mirror plane that is passing through this molecule. So whenever you pass light through this molecule, whatever bending or of light will happen here will be cancelled out by the other part. So overall you will not get any optical activity. So this is the correct option. Moving to the next one. Okay, it states proteins can be stabilized by bonds that could include the interaction of the side chain of a lysine residue with the side chain of what out of these four. So you are basically stabilizing proteins where in one of the proteins in the side chain there is lysine. Suppose in the other protein, what amino acid should be in the side chain to stabilize this lysine residues? This is basically the questions. I have options. I think all of you understand it correctly. Thank you for answering. So obviously the main thing that you have to identify here, that lysine is basically basically a basic amino acids you are dealing with. So you if you have to stabilize sorry if you have to stabilize the protein that has lysine residues, you have to stabilize it. I mean, if there is lysine residues, then it is positively charged. Like there is a, the side groups are NH2, so this will be positively charged. If you have to stabilize it, you have to give some amino acids with acidic residues, right? So out of these four, glycine, arginine, histidine and aspartic acid, glycine is not an acidic amino acid, neither arginine, neither histidine, only aspartic acid which has COOH group in its side chain as well. That is an acidic amino acid and that can stabilize lysine residues. Okay. So the correct option for this aspartic acid, option D. Moving to the next one. The most likely amino acid that would yield the above titration curve. This is the titration curve. Out of this 4 amino acid, which is the most likely to give this kind of titration curve? I have one answer.
अरे सुनो, हम लोग ऐसे ही घुसेंगे एक्चुअली। मैं और मानसी वो हमारे ग्रुप के साथ हूँ डिपार्टमेंट वाले। मैं बोलूँगी कि जिसको मैंने मैरिड दो, वो मैरिड स्कॉलर है। और हम लोग आई कार्ड दिखाएं, तो आई कार्ड दिखा देंगे।
Two answers, I guess. Okay. I would tell all of you to look at it a bit carefully to the titration curve. There are only two jumps. The initial jump is here, that corresponds to pK1, and another corresponds to pK2. That means whatever that amino acid is has only two groups that can transfer H plus, or that can react with H plus or OH minus. Only two, that means only two protonable or deprotonable groups are there. So this was your hint. Whenever we say pK1, pK2, that means there are this many numbers of groups or this many numbers of residues that can be protonated or deprotonated. So pK1, pK2 means only two residues are there, only two groups are there that can be protonated or deprotonated. So far I have got two types of answers. One is option B and another is option D. But the correct option was option C. Why so? Because as I was saying, okay, was someone answered C correctly. See, let us look at the basic structure of amino acid. I am just writing the Zwitter ionic form. And this is your R group. Okay, so this is your basic structure. At one side there will be NH3 plus, another side there will be C double O minus, and there will be an R group. So whenever we are thinking about pK values, so there is one functionality that can protonate or deprotonate. So there goes our one functionality. This is another functionality that can protonate or deprotonate, that can give rise to another pK value. So there goes our second functionality. Now if there are more pK values, then the side groups also have to have some functionalities that can protonate or deprotonate. Now first of all, look at the curve. As I said, it has only two pK values. That means in the side groups there cannot be anything that can either take up a proton or give a proton, right? Because there are only two, and we have already covered our two in case of NH3 plus and C double O minus. So basically our R is something that cannot deprotonate or gets protonated. Let us look at arginine. It cannot be arginine because in case of arginine there is an NH2 group in the side chain that can protonate or deprotonate. So that can give an additional pK value, and the graph will look something like this red one, where we'll get a pKr for the R group as well. So arginine cannot be that particular one. Glutamic acid, again in case of glutamic acid, in the side chain it has one COOH group that will again give rise to another pK value. So the graph will again look something like this, that is a two-step graph. For histidine as well, in the side chain, see there is a 5 member like in N group or NH group that can protonate or deprotonate. So again in that case also the side chain will give rise to a pK value. Only in case of glycine, out of these four, the R is hydrogen that can neither take a hydrogen atom additional or give up a hydrogen atom. So only in that case you will get one pK for NH3 or NH2 and another pK for COO minus or COOH. So for this particular question, your correct option was glycine. I hope it is clear. Let us move to the next one. Okay. The central dogma states that DNA determines an organism's traits, such as eye color or hair color. We all know that. We all get that from our DNA. Which of these sequences best represent the relationship between DNA and the traits of an organism? So these are the four traits, or you can say the four processes in a chart from that is represented here. Which of them are correct? From going from DNA to traits. I can see one answer, option A. Okay. Others also try to find a logic and answer. Okay. I have two answers. So let us look at that. So the correct answer was option A. Very good. That was the correct answer. So obviously, look at the correct option. I will just discuss the correct option, then you will understand why it is the correct one. See, first of all, from DNA based sequence, it is saying amino acid sequence. Okay, in other cases, say it is saying like DNA to protein shape, it is saying DNA to protein function. Okay, from DNA we can not directly go to protein shape, because protein shape will depend on what amino acids you are using to make that protein. So first DNA 2 you have to get to know the amino acid sequences that are there. Once you know the amino acid sequences, depending on that the protein will take its shape. Why so? If in the amino acid sequence, depending how many hydrophobic amino acids are there, or hydrophilic amino acids, or polar or non-polar amino acids are there, depending on that the protein will try to make its shape. Like if there are more number of non-polar amino acids in the present in the sequence in a particular part, then the sequence will try to shape the protein in such a way then that the particular that particular section of the sequence, suppose this is your protein sequence and this particular part has very much number of non-polar sequences involved, non-polar amino acids. This is non-polar amino acids. Now whenever this protein sequence is try to fold, it will try to fold in such a way that the non-polar ones goes in the inside part and the polar ones remains on the outside, something like that. Okay. Now whichever shape it get, depending on that, it will function. How? Because after they get their sequence and they fold it according to their most energetically favorable shape, it can be globular or it can be fibrillar proteins as well. So either they are globular proteins or fibrillar proteins, depending on that, they will function. Obviously, a globular protein, that is like a what should say likely hydrophilic proteins, they will not stay in your hair or in your nails. And similarly, of fibrillar proteins like keratin or something will not act as the roles of a globular protein. So obviously, whenever you get the shape, only after that the proteins will get their job. Okay. So after amino acid sequence, the proteins will try to figure out how it will fold itself to get minimized energy. And once it gets its shape, then the job will be assigned to it, that how it should function. And once it starts functioning, it will give our traits. Once we have our suppose fibrillar proteins for our hairs or something, suppose the keratin ones, then we can get our traits in our hair or our nails, etc. So the correct option for this question was option A. I hope this is clear to you. Moving to the next one. Okay. What effect would converting a glutamic acid to gamma carboxyglutamate have on the overall charge of the protein at pH 7? First, try to think the structure of gamma carboxyglutamate. That would help you a lot. Okay, I already have one answer. Okay, two options, but both of them are different. I can see a third option. Okay. Okay, since we don't have much time left, so I will quickly discuss this question. The correct option for this was indeed A. Why? Because if we look at the structure of gamma carboxyglutamic acid, first look at the structure of glutamic acid. COOH. And this is your NH2. Again, sorry for my bad handwriting. So this is our structure for glutamic acid. So this is our alpha carbon. This is our beta carbon and this is our gamma carbon. So it states gamma carboxyglutamate. So another carboxyl group should be added in case of gamma carboxyglutamate. Now looking at that, you can already guess that its pI value should be in the lower pH region. Why I am saying it? Its pI value would be in the region where this NH2 group will be protonated to NH3 plus. One of the COOH group will be remain unprotonated to COO minus, but the other two COOH groups should be protonated as COOH. Only in that case, overall it will be neutral. So to do that, we can already see that out of these four functionalities, three has to be remain in their protonated state. So we can already guess that the pi for this particular structure should be in the acidic region. pH 7 is what? It is a neutral region. It is not an acidic region. Even for glutamic acid, if you see, I don't remember particularly, but the pK value is, I think, sorry, pK value is somewhat around 5 or something. I don't exactly remember, I might be wrong as well. So by adding another COOH group, we are basically, the pi will be much less pH value, much acidic pi value we can say. But in our case, it states this particular gamma carboxyglutamate is at pH 7, which is at a neutral pH. So obviously under that condition, these COOH groups will not remain protonated. They will be remaining as COO minus. So obviously in case of glutamic acid, at pH 7, if there was 2 COO minus, now in case of gamma carboxyglutamate, there will be 3 COO minus, because the pH 7 is very higher than their PI values or pKa values rather. So as they are higher, as it is higher than their pK, so they will remain in that deprotonated carboxylate form. So obviously as the number of COO minus group has increased from 2 to here to 3, so the overall charge will be more negative. So this was your correct option. I think we'll have time for one last question. Okay. So it states you are given a mixture that contains glutamic acid, arginine and valine, and you subject the mixture to electrophoresis. Which amino acids migrate towards the cathode when the electrophoresis is carried at pH 7.1? So one things are important, pi values are given, so you don't have to look for it. The pH is given, and also you have to consider migration towards cathode. So these are the important points. Considering this, try to answer this question. Okay, I can see one option, option D. Okay, sorry, option D, B. Okay. Since we don't have much time, I will discuss the question. So the correct option was indeed option B. Arginine was the correct answer. Why? See, first of all, one thing we have to first think, we have to think the migration towards cathode. So cathode is itself negatively charged. Okay. So what things will migrate towards cathode? Those things that are positively charged will try to migrate towards cathode. So our pH is 7.1. So now the pi values for them are given. So only sorry, in case of this arginine, one second, sorry, only in case of arginine, if you think at pi, the pH is less than pi. So what does that mean? In case of arginine, if the pH is less than pi, in what state it will remain? Because arginine structures, so there is your one NH2 group, your one COOH group, and in the side chain also you have your another amine group. So in case of arginine, as the pH is less than pi, so this overall arginine is already remaining in its protonated form. That means everything is protonated. COOH group, COOH group will remain as COOH. NH2 group will remain like NH3 plus. This might also remain as NH3 plus because the pH is less than their pi values. Okay. So at this point, as this overall thing sorry, as this overall thing is now positively charged, so only it will try to migrate towards the cathode. In case of glutamic acid, it will not happen because the glutamic acid's pi is 3.2. So at pH 7.1, this extra COOH group is already staying still at COO minus. So it is still staying as negatively charged. So it will not move towards cathode. For valine, there is no additional R group that can uh, operate its, I mean, that can operate its acidity or basicity, so that is not an option as well. So the only one that remains positively charged at pH 7.1 is arginine, and thus it will move towards the cathode. Okay, I think with this I will stop. I have many more questions left. Those I will discuss quickly in the next class, as well as I will include the week 2 lectures. Meanwhile, if all of you have any doubts or something, you can ask or you can email in the NPTEL mail ID that I think you already know. You can mail the TS. You can directly mention my name and ask your question. I will look to the emails that are directly written for me, and I will also include the, either I will answer you in email, or if there is an interesting question, I can discuss in the next class as well. So let me stop the recording.